And thank you, Pedro and the group, for the invitation. From where I'm standing, I don't know the group very much, but I would like to congratulate you, and I think you can be very proud of what you have achieved for the last 40 years. And what I find inspiring is that you're still looking very much inspired for looking at the future with the same energy. So it's, congratulations for that. So what I thought I would do today is to tell you a little bit about our what we call basic science has transformed the way we treat melanoma patients today. And maybe two important notes here. So Evgeny already mentioned that. So basic science doesn't mean it's basic. It means, at least we like to think that it's everything but basic. We develop technologies. We try to use cutting-edge technology, but also develop our own technologies to understand mechanism of disease. And I think this is a very important thing. Another important thing is that it's not just blue sky science. So sometimes people compare basic science with blue sky science. So we always keep patients, at the center of what we are doing. And I will try to explain that towards the end of my talk. And so melanoma, I don't think you need an introduction, this audience. So basically melanoma results from the transformation of melanocyte, which is a very specific cell type in the skin. Maybe one thing that I'd like to remind you is that melanocyte derive from the neural crest lineage, and this might be important later during the presentation. So here we're looking at the very early phases of the development of the disease, up to the accumulation of cells both in the epidermis and the dermis, and the formation of what we call the primary tumor. This is actually what I guess most dermatologists are looking at and interested in. The problem is that when prevention interception fails, the disease spread very quickly to many different organs. This is an example here of a patient, and you see how much the disease can spread very dramatically to many different organs, including lung, liver, and eventually brain. And so what I will focus today is try to understand, first of all, how this disease spread. And we have a lot of work on this in the lab. The idea is that if we were to understand the mechanisms that contribute to the spreading of the disease, we might be able to intercept the spreading. But the next big question is, how do we treat patients that have evolved this very dramatic metastatic disease? And basically, when I started to work on melanoma about 15 years ago, so there was pretty much nothing for these patients. So these patients were receiving chemotherapy with very poor outcomes. And then there was a real revolution in the field through the development of targeted therapy initially for basically based on the finding that the disease is driven by MAP kinase activation. And in about 50% of the cases, this is due to a specific mutation in the BRAF gene. And so pharma companies developed molecules, as you know, these inhibitors that target specifically this mutated form of BRAF. And this led to dramatic anti-tumor responses in these patients for the first time ever. Now, this will come with caveats, and I will show you that in a minute. Another major breakthrough, and again, it's a breakthrough that comes from the work looking at, people looking at melanoma biology, is the development of this immune checkpoint blockade. So the realization that cancer cells are able to hide from the immune system by turning on checkpoints. And so hence the development of these inhibitors that basically block these, the immune checkpoint. This was a major breakthrough in the field, so you've probably heard about anti-PD-1. This is now not only used in the context of melanoma, but this is used broadly in many different tumor types. So these major breakthroughs in the field came together with a major improvement in overall survival, as you can see here on the right-hand side. So now we're talking about long-term survival for many patients. When it comes to targeted therapy, as you will see, these responses are very dramatic, but only transient. But when it comes to immunotherapy, immune checkpoint blockade, patients respond long-term, and some of them are considered cured because they've been followed for more than 10 years. So this is the great news. So you can see that basically we went from poor outcome to almost 50, 70% of the patients responding long-term to this treatment. It's like major changes in the landscape over about 10 years or so. So when it comes to targeted therapy, as I mentioned, so we target the MAP kinase signaling pathway. Now we use a BRAF inhibitor in combination with the MEK inhibitors. And again, you see these dramatic anti-tumor responses, but the problem is that tumors reappear relatively quickly. And of course, one of the big questions is why is that, and can we prevent that? And so, the first trivial explanation for why these tumors regrow is that these treatments do not kill all tumor cells. They always leave a number of cells behind, which we refer to as the resistor cells, or these drug-tolerant resistor cells. And the question is really why these cells are emerging, and this is simply because we have come to realize that tumors are extremely complex, and not all tumor cells are created equal, right? So in response to treatments, since the cells are not created equal, they will respond differently to the treatment. So some cells will die, other cells will just stop dividing, while other cells may even continue proliferating because they're completely immune to the treatment. So this is what we call intratumor heterogeneity, and the problem that we're facing when trying to treat cancers with any given therapy, actually. And so up to recently, it has been very difficult to dissect this phenotypic diversity. Technologies would not allow us to interrogate every single cell in a tumor. But thanks to the advent of single-cell sequencing, and Yevgenia already mentioned that this is now possible to take a tumor, make single-cell suspension, and then look at the transcriptome of every cell or every individual cell in these tumors. This has been a revolution in the field that was introduced about 10 years ago. So we have collected a lot of melanoma lesions from patients as well as from mouse models, and then we leveraged this single-cell RNA sequencing and look at the diversity that one finds in melanoma lesions. And to make a long story short, we find that melanoma cells exist in a continuum of what we call cell states, and they move along a dedifferentiation trajectories. So we, based on transcriptomic analysis, define 4 different cell states, one of which we call melanocytic, because these cells express basically the same gene expression programs found in melanocytes, in normal melanocytes. And then cells move along this dedifferentiation trajectory. They shut down expression of these melanocytic genes, and they turn on this more embryonic neural crest-like cells, so the cells of origin of melanocyte. So they become more embryonic-like. And eventually some cells can dedifferentiate almost completely and lose their melanocytic identity, and they become like fibroblasts, they look like fibroblasts, and we call them mesenchymal. And so obviously this is what we could observe by looking at tumors that were drug naive, or non-treated tumors. And of course, the big question was, what happens to these persistent cells? Can we use these technologies and understand the biology of these persistent cells? And naively thinking, if we were to understand the biology of these cells, we might be able to target these cells and prevent the development of resistance. So we've developed a number of models to actually look at these persistent cells. As you can imagine, it's not easy to go and fish out these persistent cells in patients. So we developed PDX, patient-derived tumor xenografts. So we basically take tumors from patients that respond well to the treatment, implant it in mice, and then generate models. And then we treat these models with the standard of care treatment, so a combination of BRAF and MEK inhibitors. And then you see these tumors regressing very quickly, like we see in patients. And then we can start fishing out these persistent cells and then performing single-cell RNA sequencing and ask, what are these cells? And to make a long story short, we find that the cells that persist the treatment often move along this dedifferentiation trajectory and become more neural crest-like or more mesenchymal cells. These are the cells that seem to persist and evade the therapeutic effect. And so because of that, we developed a number of strategies to target these cells or to prevent emergence of these cells. Maybe I should also mention that, in fact, these cells can emerge through 2 different mechanisms. First, they can be enriched because some of these cells are actually present before treatment. And because they are resistant to the treatment, they are enriched over the course of the treatment, because the sensitive cells die, and those cells don't die and basically persist. And then, but importantly, looking at these cells in a dynamic way and using very fancy technologies like barcoding technologies, we've been able to also show that melanoma cells can actually acquire these phenotypes. So they are able to adapt to the treatment and turn on these more mesenchymal-like features. So cancer cells are able to evade the treatment in a non-genetic way. We call this cancer cell plasticity, the ability of cells to adapt. So there's really two different mechanisms through which the cells can emerge. And so we develop strategies that, one, block the ability of cells to adapt on the one hand, and strategies that are targeting these already dedifferentiated cells, because basically we're thinking that targeting these dedifferentiated cells might not be sufficient. We might need to also find ways to block the dedifferentiation. And so and I skip you, the details, the molecular details, but we found a number of ways through which we could actually do that by targeting the some epigenetic modifiers, for example. And these are survival curves of preclinical models. So you can see here, for example, the blue curve is when we treat our PDX model with the standard of care treatment, BRAF MEK inhibitors. Eventually, mice die because they develop resistance. And now if we combine these drugs with a number of other pharmacological agents, we now see that basically we delay time to progression. So the mice are doing better and better, and eventually we see that there is potential benefit based on this preclinical work. Now, we were quite excited by this data, but as you can see, we cannot even cure these mice yet. So eventually all the mice develop resistance ultimately. And the reason for this is that dedifferentiation is only part of the story. There's actually more persistent cells that we initially thought they were based on the first type of analysis of single-cell RNA sequencing. And now, basically, we're still busy trying to understand what are these other melanoma cells that resist the treatment and try to devise therapeutic strategies against these cells. And just to give you an idea of the complexity we're dealing with here, now we are now able to look at these persistent cells from patients, from lesions taken from patients that are responding to the treatment, thanks to the neoadjuvant setting now that we use for melanoma. And basically what we concluded is that pretty much every single lesion that is regressing look very different under the microscope or using this type of technology. So in a way, the cellular makeup of this residual disease is very different and patient-specific, indicating that we have still a long way to go to understand the biology underlying this emergence of these cells and treat patients in a personalized way. I will come back to that in a minute. Now, the other thing that in the fields I mentioned we are very excited about is the fact that we can treat patients with immune checkpoint blockade, basically essentially anti-PD-1 treatment or therapies. And in this case, as opposed to targeted therapy, the problem is more intrinsic resistance. So when patients respond to the treatment, they respond long-term. There is rarely development of resistance to these treatments. The problem is that some patients do not respond upfront to the treatment, and we call this intrinsic resistance. And here, of course, the questions are, can we identify these patients upfront? Because all these patients receive the treatment and some will not benefit from this treatment. And of course, more importantly, can we find ways, or can we understand why these patients are intrinsically resistant and intervene and find combination strategies that would overcome this intrinsic resistance? And so to address this question, what we decided to do is to initiate a prospective longitudinal clinical trial in our institute and collected samples from patients that receive immune checkpoint blockade. So we collect samples before treatment and very early on treatment, 2 weeks on treatment, so before the second infusion, actually. And then we process the samples with this, again, fancy technology, single cell and spatial transcriptomics. Just one note about the spatial transcriptomic methods. I don't know if you have been exposed to that yet, or if you realize that this is really like a revolution in the field. So beside now single-cell RNA sequencing, we can actually look at almost the entire transcriptome of individual cells on a single slide, right? So we can, here today, we can look at 50,000 genes in a single cell on a slide, and we can look at millions of cells all in once, and we can look at the position of these cells in the tumor. This is a revolution in the field, not only for cancer, but in biology in general, because now we understand, our tissues are formed, and this is really helping us now to gain major insights into tissue architecture and cancer architecture. And what you see actually here is a melanoma lesion that we processed with this technology, and we are now staining or coloring, because this is a pseudo-colored image, the different melanoma cells with different colors. And what you see is that they're not randomly distributed. There is some kind of an organization. For example, you see these red cells here that tend to occur around blood vessels, and then there is yellow cells that tend to be away from blood vessels. So there is some kind of architecture. There is a spatial distribution code we're trying to understand. Now, with these methods, again, we looked at these lesions from patients before treatment and early on treatment and asked, what features can we identify in these patients that do not respond to the treatment? And again, what we found is that this more mesenchymal-like, this dedifferentiated population accumulates on treatment in these patients that will not respond to the treatment. What we were very surprised of is that this population in this patient is extremely rare. Even if it's only a few percent, if that population only represents a few percent of the entire population, if that population is present, is extremely strong indicator of lack of response. And so this suggested that, again, this ability of cells to move along these dedifferentiation trajectories is a way through which cells and tumors find an escape route to the treatment. And basically indicating that these more mesenchymal cells are, again, extremely good at hiding from the immune system. And the question is, why they do that? But again, the idea that the cells are able to differentiate is a major escape route towards therapy exposure, and now not only targeted therapy, but also immunotherapy. So mechanistically, we identified a gene regulatory network and a molecule called TCF4, which is a major player in the ability of controlling the ability of these melanoma cells and these mesenchymal cells to evade the immune system. So this transcription factor not only instructs the cells to become more mesenchymal, they differentiate it, it also instructs the cells to shut down expression of these melanocytic genes, which are often recognized by the immune system. But even more profoundly, this transcription factor suppresses altogether antigen processing and presentation. And so these mesenchymal cells basically don't present any antigen at the surface and are therefore not recognized by the immune system. So this allowed us now to develop pharmacological strategies to target this pathway. So we first showed that genetically, if you use different genetic approach and targeting this transcription factor, you can enhance response to immunotherapy. And then we also developed therapeutics, pharmacological and therapeutic strategies that target this pathway. And quite strikingly, if we use now different preclinical models, so these are mouse models that do not respond to anti-PD-1, so they are engineered so that they don't respond to anti-PD-1. Here you look at tumor growth, and then you can look at response to anti-PD-1. Basically, these tumors do not respond to anti-PD-1, but if we target TCF4, you see this pink curve, this dramatic tumor control that we find in this model. So again, very exciting. It's preclinical work, and now the question is, can we bring this closer to the patient? This is really, for us, something that we have been thinking a lot about for the last few years or so, because we're pretty convinced that some of the combinations that we have developed in our preclinical models can really benefit patients. But how do we bring this to the clinic? The first problem that we try to solve is addressing the challenges of what we call interpatient variability, and the fact that every patient might benefit from different type of combinations, right? So different combinations might benefit different patients. And the problem is now can we treat these patients in a rational way, understanding the cellular makeup of these tumors. Now, I've told you that we've been using this single-cell RNA sequencing or spatial transcriptomic methods. These methods are extremely expensive, so everyone knows this really well. So implementing this in the lab is possible. It requires a lot of efforts to get funding, to think that we will use these techniques in every individual patient in the clinic, because we're talking about thousands of euros for every single lesion. Right? So the first question is, can we develop a very simple, cheap method to monitor intratumor heterogeneity in patients? And so we decided to think about this, and we were quite inspired by this definition of phenotype that was first introduced almost a decade, 100 years ago, which is the observable trace or appearance of an organism, right? So we decided to actually look at our melanoma cells, because right now we're characterizing these different subpopulations based on transcriptomics, but the question is, can we actually find other ways to characterize those cells? And we thought maybe just looking at our cells will help us to go about this. And looking at our cells using very fancy microscope, we realized that the cells that we group into different subclusters look quite different under the microscope, indicating that morphometrics may actually be used as a tool to discriminate between these different melanoma subpopulations. And here we team up with a company in the US that developed methods that allow us to look at single individual cells and look at morphology of the cells. So here we are looking at many different parametric measurements. So looking at size, volume, colors, and then we basically can interrogate our tumors. So here we basically fed this device with more than 20 different melanoma cell lines that we have in the lab that are well characterized. And basically the breakthrough is that we realized that these different melanoma subpopulations that we had classified as melanocytic mesenchymal basically exhibit different morphometrics. And it took us now quite some time to define a minimal set of parameters that allow us to discriminate between these different melanoma subpopulations. And just I'm going to make a long story short, we realized that cell surface is a very good discriminator between the melanocytic and the mesenchymal cells. So the mesenchymal cells have a very irregular cell surface that we can easily monitor by FACS analysis. And then the other parameter is cell volume. So we can look at cell volume again by FACS by using a very simple method, which is basically by looking at forward scatter width. And with that method, we could separate very nicely the transitory and the neural crest-like cells. And basically now what we can do with a very simple FACS method, we take our melanoma cell lines, and then we can find that we can very easily separate the different melanoma subpopulations we identify by transcriptomic methods. So this method we call origami. So we simply define the position of a cell into this two-dimensional space as what we call a morphocell state. Now this is all nice and cool as a tool for looking at intratumor heterogeneity in tissue culture, but the question is, can we implement that in the clinic and by looking at biopsies? And so for implementing this technique from biopsies, what we needed to do is deplete cells, deplete these tumors from immune cells, because obviously when you take a tumor, you make single cell suspension, you have immune cells, you have fibroblasts. So we needed to deplete these cells. So we found a way to deplete these cells and then proceed with our surface and volume measurement. And in parallel, for the same lesions, we perform single-cell RNA sequencing and find that indeed these methods capture beautifully the intratumor heterogeneity that we see by single-cell RNA sequencing. The only difference is that it takes about 20 minutes as opposed to 1 week, and it takes about €1 as opposed to €1,000. Right? So there's huge difference in cost and time by implementing this technique. So we are now implementing this in the clinic. When we treat a patient for melanoma, we take our surface and volume measurement, and we think about ways through which we can deliver a better treatment according to this very simple measurement. So all this to say that I think this value at, of course, using these very fancy technologies, but also developing very cheap and simple alternatives that we can implement in a clinical routine. And of course, we are very engaged into that. And then the final thing is that how do we bring these combinations that we find in the preclinical setting in patients? This is extremely difficult, given that there is no strategies and regulatory paths for drug testing or for combination testing in patients. So for us, it has been a major drawback, and we had to invent something and be quite creative. And we teamed up with a company called Presage that has developed a tool that allows you to do intratumor microdosing of drugs in patients. And so here, basically what this tool allows you to do is to inject and microdose up to 8 or 16 different drugs in tumors from patients. And because we're talking about microdosing, there's no systemic exposure. You can even deliver non-FDA approved drugs in tumors from these patients. So for us, it's a major leap forward. So we initiated the first clinical trial in Europe of that kind. So basically, melanoma patients come in, they receive anti-PD-1, so systemically, and then intratumorally, locally, we deliver up to 8 different drugs, drugs that we developed, or combination that we developed based on our preclinical work. Now we can test the efficacy by looking at molecular and cellular response locally around the drug diffusion area. And we have done now a couple of patients, and we're very excited about that because we actually see that some of those patients that do not respond to systemic anti-PD-1 may benefit from some of the drugs that we bring in locally. And the idea is now, can we use this method to personalize this combination treatments directly in patients? So all this to say that, we're not only doing blue sky science, we try to bring some of our combinations to patients. And so finally, a note of enthusiasm. So I mentioned, our work on targeted therapy, immune checkpoint blockade. Patients are now responding long-term. And the excitement in the field is that, it doesn't stop here. There's many, many more innovations that are now being tested in patients. And we are very optimistic in the future, as opposed to, 15 years ago. For those patients, there was nothing, and now we're really moving forward. And so all this to say that I think it's fair to say that basic science. So, for example, looking at the etiology of the disease, finding BRAF oncogene mutations, identifying these checkpoint blockades has really transformed the clinical management of melanoma patients today. Importantly, the work that was done in melanoma benefited not only melanoma patients, but way beyond that. Yet, too many patients still do not benefit from some of these treatments, and we are actively working in the lab to try to overcome this problem. And with that, I just want to thank you for your attention and thank my group, obviously, and hope that I could interest you in some of the work we're doing. Of course, it's very much upstream what you are doing, but I think these two worlds need to collide, and I think for that I thank you for the invitation and for the opportunity to present some of the work that we're doing. Thank you.